Hey guys. Let's see. I'm having some extremely serious technical difficulties right now. I'm home today. I got really sick overnight. I haven't been asleep at all. I've just been in the bathroom. So I'm up at home today and I'm trying to pull up your PowerPoint for the microscope stuff, and I'm looking on my USB. I work off of USB for this reason, so that I can go between, and it looks like I worked super hard on this freaking presentation, and it is not on my USB drive. It looks like it's saved probably entirely to my actual laptop at work. I wasn't planning on being sick today, so I don't have my laptop from work here. I was planning on being at work. So I didn't save it on my USB drive, and I'm just trying to see if I just saved it somewhere weird on the USB drive. So give me a minute, and if I cannot find it, um, I mean, I don't have any other PowerPoint. That's the only PowerPoint I have. And so I will pretty much just be having you guys watch a video, which I was going to have you watch anyways, but it'll be just that. And I can't put one together as we go, right? So I'll have to pull that up. So I have a roster. It just didn't save it at all on here. It's just all on the freaking hard drive, as in it had to be. I don't know what it is about the past couple of weeks, but it's not just me. It's from everybody on in the science department. Everything has just been screwed up. Search. It doesn't have it. Sometimes it saves things to the cloud, but I don't know how to access that from home. I mean, I have the VPN. I just don't know how to. I've never had to try to get like that. Sorry. I'm sure you guys aren't crying about it though. When I get back to work, I will just upload that PowerPoint, like the actual PowerPoints on there, so that you guys can go over it. So it has information in there that I feel like is more helpful than just like the handouts. I can at least pull up the handout and we'll kind of go over the content that's in it. Let me get and I'll share my screen and everything like that. So at least we can do that and get roll going. I've never had to teach like all virtual labs ever. I came after COVID. So I've only been in person labs. I've had a few that I've had to like, for weather or whatever. I have like mini labs that I've recorded, and that's what I'm going to show you guys today is one of those. But geez, man, this is just been something else. And then, okay, let's see. So you guys are 47. Let's see. That is not known, that is Discord. Discord. Okay. Let's get this. See me looking like I'm about to die here at home. Share screen. Okay. So, all right, so this is exercise 2. It's dealing with colony morphology, simple stains, and how to use microscopes basically. And I know that you guys have seen a microscope, and I know that you probably know in general how to use a microscope. A lot of the bacteria that we look at in our microscopes in microbiology, we're looking at the highest power, which like generally when people come into my lab have never actually used the highest power, which is the one that we use oil immersion with. And so if you've never had to add oil to it, whenever you're using it, you have not used the highest power lens. All the microscopes that we have at OSU OKC all work that way. So if you have been in physiology, whoever you took it with, but typically, I would be talking about Dr. Shearer. If you have taken physiology lab and used the microscopes in there, our lab in microbiology, we use the exact same microscopes. So it'll at least be familiar if you have used that. Same parts and everything, but we spend almost all of our time exclusively in that oil immersion lens. And you absolutely 100%, once you get into those, do not need to be touching the coarse focus, the big knob. So yeah. Sorry, I'm trying to take I'm talking with this at the same time, take attendance. So anyways, because it'll crack the lens, potentially damage the lens, which is like super, I mean, again, if you guys understand how expensive it is to fix something like that, super expensive, or just crack your slide itself, which they are actual glass. So you could be shattering glass potentially all over people who are putting their eyes very close to it. So let's not do it. But yeah, so we'll talk about how to use a microscope a little bit in a moment, but there's that. So we would have in the last lab done our little quad streak and turn thing, right? Streak and turn, streak and turn, streak and turn. And in that fourth quadrant, if we were to have looked at it, you would have had individual little dots, colonies, and each colony in from one cell, right? So we could pick one of those to create a pure culture from if we wanted to. But first, it's not all about just only picking like the one colony and then growing it in, looking at it in stains, growing it on different media and looking at changes or not changes, and then using that info to determine what it is. The actual appearance of what the bacteria look like on the plate, like the colors of the colonies, the sizes of them, what their edges look like on the plate, and all of that can be used as identifiers as well. For example, with Staphylococcus aureus, the colonies have a golden glow to them. Not a literal like glow with light, but just like a golden glowing color to them. And so that golden glow, that is why it's called aureus, like the aura. That's why it's named what it is. And its first part of its name is actually another thing we're going to be learning about today, which is like the arrangement and the shapes of the cells themselves, which we'll go over. But yeah, I'll probably just like, as we're going, Google some of this stuff so I can show you what I'm talking about, so that it's not just all me saying things and then this word's on the screen and whatever, since I don't have my PowerPoint. We're having this trouble with the student who is not able to log in, so I'm still just trying to knock out this. Like, let me do this just real quick. There's not enough of you guys for me to be taking this much time doing this. Okay, Donna, Marissa. Nadia. Sharon. Missing Destiny. Oh, no, Emma's here. Sorry, Emma's like, I see it. So we're missing Katie. Who we have yet to see. We're also missing Destiny, but she's just having, she's the one who having problems getting her on. So that's why I want to mark that off so I can just email her. She can't log into Zoom for some reason, but that's not enough information for me to help. So anyways, so let's talk about colony morphology, shall we? There's terms, and I can't like draw on the screen. I apologize, since it's like just my screen. It's not a PowerPoint. So you know, whatever, but it's not ideal at all, and that's fine. Colony morphology. Morphology of something is just talking about like the shape and how you like what it looks like to you. So don't worry too much, like when you're reading these terms, don't try to fall into the trap of big word, can't understand it. Try to break it down into parts that you would. And to me, morphology, ology, study of morph, like if you're going to be morphing into something, shape and everything like that. So it literally is that the different shapes, study of the different shapes of colonies. So that's all that word means. So the terms that we want to focus on are dealing with over here on the left side. Oh God, I can't even like, where it says like shape and margin, surface, texture, elevation, and then color and translucency versus opaque. All of those are like categories where you would pick terms from. So if we were looking at your bacteria, how would you describe your bacteria, which I know we don't have real bacteria, and on our PowerPoint slides, which don't exist apparently, I had put together examples that I wanted you guys to work from for like what yours would look like, what another tables would look like, and what your slides would look like for the microscopes that we work with. Anyways, sorry, I'm trying to deal with that student who can't log in. It's hard for me to help you guys too. Like if you are having issues, like I can't log into Zoom, and that's all you tell me. I don't know if it's like, did you do the SSI thing? Because you have to do that every single time. Like even I have to do that every single time. And are you logging in with your OSU, you know, logins and all that stuff like that? Because if you don't do that stuff, then you won't be able to access the Zoom, unfortunately. But I don't know what's going on. So whatever. Shape, easy enough to think about. the shape of the actual dot that you're talking about. So if we're looking at the colonies individually, then what's the shape of each colony? Are they, because they're not all round. Y'all probably all think it's all just gonna be little dot, white little dots, and that's all you see, and that's absolutely not true. Sometimes they can be different colors and have different like wiggly edges and everything like that. So the actual shape of the colonies themselves, a lot of times, a lot of them are just round, but it could be irregular. And the more common ones that you can see, those tiny little dots that are super, super tiny, we call that punctiform. So that's why that's underlined, because that's a word that people don't usually know. So just tiny little dots all over. All right, the next term is margin. So margin is referring to the edge around the colony. Is the edge like, so we know it might have a round shape, but in its general round shape, is it an entire or a smooth edge? So those are both kind of the same thing. It says smoot. I understand. I never corrected it. Everybody asks me what smoot means. It means I don't know how to spell smooth. This is what it means. It was just a spelling error. I hand typed all of these worksheets. So sorry, there's some spelling errors in there, but smooth. You guys obviously, in case you haven't figured out, I don't really have anything in this course where I have to grade you on your spelling of something, because I hate that crap. I think if you get the concept and you know what the word should be, if you are misspelling it by a letter, you should not get counted off for those points. So that's my personal opinion about it. And that's how I teach my class. You know, I know that's not how anatomy runs, but that's how it runs in my class. I just don't care about that. Is that the important part of what we're teaching? Not in my class. So, yeah, welcome, I guess, for that a little bit. There, right, so lobate, it would look like it has, sorry, did you have a question? Probably just
Sorry,
unmuted.
no, sorry.
Okay, you're okay. Okay, so Destiny's here with us. Glad you made it. Sorry that there was an issue for you. Gotcha. Attendance. Okay, so yeah, it would look like lobes, and I'll see if I can find some pictures. I'll just look them up like when we're done going over colony morphology, to give you some examples, because they'll probably all be all together and we can just look at it that way. So I don't want to go to it just yet and keep going back and forth. Destiny, since you're just joining, you get to be the last one to receive the news. I made a very intricate PowerPoint for this, and I'm clearly not at my office right now. I'm homesick, and my best friend today is Pepto-Bismol. So, and I have electrolytes in stomach soothing tea. It's that bad. So, but yeah, so I'm home, and we're hoping we can make it through this, but I didn't save my lecture on my USB like I always do. So I should have known I was going to be sick. It's stuff like that, you know, like you're like, oh yeah, I was sick. I forgot to save it on the right drive or whatever. Which, by the way, if you guys haven't tried those electrolyte drops that are like just electrolytes, they have no flavor in them at all. Electrolytes are salts, so they are salty in and of themselves. But if you add the amount that you're supposed to be having when you need electrolytes, like when you're working out or when you're sick or something, it shouldn't have a saltiness too much in your drink. You might slightly taste it. You can put it in like anything. So I hate Gatorade. So I don't like sweet things really too much. Just, I don't know what it is. I don't like it. But anyways, I have some stomach soothing. It's like ginger and whatever. I don't know what even is in it. It's a lot of ginger probably. But yeah, it's like peach flavor. But there's no sugar. I don't take sugar in my tea. So, but I take electrolytes in it today. And I don't taste them, luckily, because that would be gross with that flavor. But yeah, so that's what we're living on today, that and Pepto-Bismol. It's been a rough week, you guys. So margin, lobate, it's like lobes. Like if you literally just had like almost like flower petals around it, and then sort of like joined in the middle, as opposed to being like connected at little points. So it's like just lobes around. So that's lobate. Filamentous is just having a lot of filaments coming off. Like if you would think about like at a center, just all these little hairs coming out of the center. And if you have a lot of branches coming off, looking sort of like roots, that is rhizoid. We see these shapes a lot, filamentous and rhizoid in fungi, but bacterial colonies can have these shapes too when they're growing on the surface of plates. So, yeah. So we'll see pictures in a moment. I promise I'll look some up for you guys so you can see the right thing and know that you're looking at the right thing. Okay, obviously these are in your lab manual as well. That's probably a good place to look at it. I mean, opened up Top Hat today. That's my bad. I'm sure you all did your pre-labs, but I am planning on looking at that afterwards. Just wanted to get this knocked out of the way. I thought it would be easier, but surface. The actual surface of the colony, whether it's smooth or it has a rough texture to it, or if it has a wrinkled. Some of them look like they have almost like a layer on them that got accidentally wrinkled up and gross. But yeah, that's sometimes what they can look like. Texture is the actual texture of the colony itself. If you were to like poke at it and smear it around a bit, what its texture is like. So whether it's just a moist texture, mucoid, which is like mucus, you know how it like sticks together and be sticky and everything like that. Butyrus, it has like a buttery texture and so really smooth texture to that. That wouldn't be super too wet like the moist. Dry, what it sounds like, it might be kind of flaky and everything like that. And then whether it's shiny or whether it's dull, all of this stuff applying to the texture. Then the elevation is talking about like if you were to look at the colony from the side, so you would have the surface of the auger, and then if the colony is growing on it, what it looked like from that side. So is it just flat completely? It doesn't seem to have anything characteristic about it as far as how it's raised off the auger. Is it convex, which is just round, like what we would expect, right? Just like round. Okay. Fulvinate is like very convex, almost like you could think of, I won't say like a mountain, but like a big hill. Okay. Umbonate is like a fried egg, so it has flat and it's raised in the middle. Crateriform or crateriform, I don't care how you say it. Again, the concept's more important to me, but that would be like a volcano shape. So it comes up high and then it kind of dips in the middle. We often see that with those wrinkled textures simply because, you know, we would associate that with, I don't know, they do that. I don't know how to describe it, but they look like a volcano because they've got the texture on the side too. So they tend to have that wrinkled texture on the surface. And color is pretty straightforward. I mean, what color is the colony? It can be all sorts of different colors naturally. And then we have translucency versus opaque. Can you see through the growth, or is it like completely opaque, solid in color? But yeah, so that's how you can very first start to identify your bacteria, how they look like on the plate. That term is colony morphology. When we get to the end of this lab project that we're going to be working on, when we get to the end of all that, one of the characteristics that it asks you to detail about your samples, your gram-positive and your gram-negative bacteria, is going to be your colony morphology. There will be, and eventually when we get to growing things, you'll see it, but there'll be plenty of opportunities to assess your colony morphology. So it doesn't have to be this one experiment that we're not going to get to. The whole second half of the lab is going to be growing stuff on different kinds of media, and you can assess your colony morphology on any of that. So at some point, I'll remind you guys about this. Remember colony morphology, the one we didn't have a PowerPoint for, and you had all these terms on how to describe your colonies. You might want to go, make sure you go and get that information about your organism and write it down about what you see about your organism. So, when there's people out the door. I mean, I'm sure everybody does. I feel like such a millennial right now. Okay, so what did okay, Alexa just said something. What she said. It's just a mailman. My dogs are freaking. Anyways, yeah, so before we get into the microscope stuff, let's go ahead and pick one of these tabs that I'm not sure what they are. this one looks good. And let's look at some colony morphology stuff. Yeah, it really is going to be like this today. Images. Okay, so this is a pretty good one. We look at the side on the left over here. We can see all these different colors and all these different shapes. So I'm not going to make you go through and necessarily name all of these right now, but you can see colony 6 has that kind of characteristic egg shape to it. So it's umbonate, right? So that's what we would apply to colony number 6 here in the middle. And you might even say like, oh, look, it looks like it's opaque. It has like a almost golden orange color to it. I mean, I can't exactly poke at it to look at the texture, but I'm guessing it looks a little bit mucoid to me. It is shiny, it's not dull. It seems to have smooth edges. You could say, I thought it was a little bit irregular, but yeah. When you guys are writing these terms down to describe your own growth on the plate, it doesn't have to be, it's more for you to describe it than it is to hit the exact ticks of the terms, if that makes sense. So whenever you describe your own growth on your own plate, I want you to try to use these terms that apply appropriately in microbiology to whatever you're seeing on your plate. Don't just say, it had a buttery appearance. Yes, write butyrus. If you're writing buttery appearance and you're not using butyrus, I want to kind of look at you like, okay, are you really, describe it, that's great, but are you going to get all your full points if we actually introduced that term? Well, I don't know. So do your best to describe it. But if you feel like it isn't like completely smooth, perfectly round all the way around, it has some irregular aspect to it, you could put that on here as it's time for the telemarketers to call. I accidentally picked up the phone. Don't do it. Never answer the phone if you aren't sure who it is. I mean, I'm completely serious, simply because I get about probably 30 phone calls a day now from spam and telemarketers. It's different phone numbers every time. So like I can't block them. And now I just have to just like silence it and let it go so that they will eventually stop calling me, because I answered one phone call by accident. And telemarketers, like they sell your phone number to other telemarketers. So this world, man. Okay, but anyways. So that one doesn't have any other like terms on it. So let's see. These are pretty good little charts. I just want to get too complicated because you guys don't know too much. This is a good one. So like punctiform, the little dots. This one's circular, or you could say round. That is fine. Filamentous, it's growing out. Rhizoid, we've got branches. That's the difference between these two. Branching off like roots coming out, or filamentous growing out without branching off like that. Irregular edge. Spindle, I guess, would be like if it looks like it has like elongated colonies that are almost stretching out. Elevation, now you're looking at it from the side, whether it's flat, whether it's raised at all up there, whether it's convex, so it has a curvature to it, or pulvinate, really curved up, or umbonate, which is looking like that fried egg shape. And then the margin, this is talking just about the edges of the whole colony. So whether you're looking at it from the side or from the top or anything like that, so just the edges of it in general. Entire, so just meaning it's nice and uniform all the way around. Undulate, I guess you could say, is kind of irregular. I'm fine with irregular. These aren't our terms. These are lobate. You see how we've got all these like lobes coming out of it. It reminds me of flowers a bit, but they can be even bigger lobes than that. We did not learn this term, so don't worry about that one. Filamentous, again, and same concept as up here, and then curled and all this. So these are good terms for becoming familiar with these. I'm not going to look for every single term that we ever learned on here, just because it's going to take me a while to sort through these, and that's half the problem of putting those talks together. But here we have just really quick. See how this has a dip in the middle of it? So I mean, I'm not going to lie. I'm just going to say it because we're all adults here. This looks kind of like a cervix to me. It does. So, you know, it's got this kind of crater in the middle of it where it's dipping down in the center. You have the shiny, almost mucousy appearance to it and everything like that. So this is a good example of crater form that doesn't look like straight up volcano, but it still has that dip in the center. So that's kind of what that's about. But yeah, so it says this one's convex to me. That is very convex to me. So, I mean, it depends on how you want to look at it, I guess, but yeah, fulminate. But anyway, so that's the idea with the colony morphology. Let's get back to that. So those are terms that you would need to know on that. That's the big picture, right? That's like macro aspect of microbiology. We don't need a microscope for that stuff. So that's stuff you can already learn about your bacteria without using a microscope, and just by growing it on regular media. So let's move on to the microscopes. Obviously, microscope is there to help enlarge your image of your microorganism. That's the whole point of the course. And we're going to be using it to make that look bigger. So I have a picture of our own microscopes. Let's just find one, shall we? It's actually someone that's labeled. Okay, so when you're looking at a microscope, you're probably looking into, let's go from the top, right? You're probably looking in from the top. It's this piece up here at the top, the eyepiece or the ocular lens. The lens itself, it does magnify in the eyepiece. Usually on most microscopes and on the microscope that we're using in our labs, it has a magnification of 10x. And what that means is that whatever you see through that specific lens will magnify whatever it's magnifying by 10 times, okay? Every time you introduce another lens, you will be multiplying. So we don't start out with your image. It doesn't start at your eyepiece, right? Which is what you're looking at in the microscope. It's down here on the stage. And so the first lens that it's actually going to be going through are these objective lenses down here, which you can switch between, right? And so your actual slide will be sitting on this platform here. This is the stage. Our stage on our microscope looks more like this. I know it's not labeled, we're going to go back, but ours looks a lot more like that. Not exactly, but it's more similar. And we have these little like, I don't know, pinchers at the side that hold your slide in place. It doesn't hold it in from the top. So those things will hold your slide steady onto your stage, so that whenever you move your slide around, it shouldn't be. No. Let me see if I can. Okay, so this is more like what we will be working with. If not exactly, this might be our microscope, honestly. This is a drawing, I'm aware, but a drawing of our microscope is very close. So you've got the little stage clip thing that's going to hold it in and cinch it in. So you make your slides, you put it onto the stage, like that's just setting the stage, all of your images put onto the stage and put into place by the stage clips that'll hold it on there. If you need to move your image around, let's say you put your slide down onto your stage and you don't like where it's positioned on your stage, like it's not even, the light isn't even shining through your spot on your slide. It's like through the side of the slide or something like that, and you need to move it. You do not push it around on the stage, okay? That could break the microscope. Please do never do that. Use these little knobs on the side over here, and it'll be on the same side as you see on this picture for you on the right side. And those knobs, so those like a top one and a bottom one, will move it either left to right or front to back. So you just turn those knobs and they will move for you. These are called the stage controls. And they're probably one of the more important things when I'm dealing with people who are just trying to push their slide around on the top of the thing, okay? If you're having a problem remembering how to move your slide around, or if you're having a problem remembering how to move the stage up and down, because when you get a microscope out, that stage should be as far down as it can go, so that, you know, when you are moving it around the microscope, literally, you aren't, you know, have things close to the lenses on the objective lenses and everything like that. You have space there and nothing can get damaged. So you usually want to store your microscope with that stage moved all the way down. The way you move the stage up and down, I told you how to move it right and left and forward and back with those knobs at the side, right? The things that hang down, they're knobs that hang on the thing hanging down. On the side of the microscope, you can see quite clearly this thick knob with a little knob kind of sticking out of it. That's the coarse focus and the fine focus, respectively. So the big one is the coarse, just like if you had coarsely ground salt and finely ground salt, right? Big is coarse and fine is small. So that's the idea. You will use turning those knobs to move your stage up and down. That is what those knobs do. So they don't do anything with the lenses. A lot of people get confused with that. Those knobs are literally moving the stage up and down. So that is why I tell you, like, once you have your thing ready to go on your higher objective lenses, you do not need to be touching that coarse knob. It's going to move your stage up and down in bigger movements. The fine focus moves the stage up and down. It's the same stage, same movements, but just finer amounts when you're turning. It's slower. So we don't use the coarse anymore. So that's why we do it that way. So you get your microscope out, you grab it by the arm, by the way, which is that back, looks like the spine of the microscope, what they call the arm. And you put your hand under the base, which is just literally the base of it. And then you lift it that way, always two hands, okay? So you don't drop it. Then, then put your slide on there. You'll want to turn your light on because this whole thing is going to magnify how light is passing through your specimen. So there's the switch at the bottom. I think ours is on the back. And then there's the brightness adjustment. So this is going to adjust the amount of light coming out of your light source or illuminator, same thing. Okay, it's just a light, like a light bulb's in there, and it's like a dimmer. Okay, that's all the brightness adjustment is. Shines light up through this structure underneath the stage that is called the condenser. The condenser itself is going to focus light onto your specimen, which is sitting on the stage right above it. So that's the condenser. You can even control how much light is going through the condenser by a little lever on the structure underneath the condenser, or as part of the condenser. It's called the diaphragm or the iris diaphragm. And it's like the iris of your eye. It goes tighter or wider based on whenever you're kind of sliding your little slider. And that will adjust how much light is passing through. That versus the brightness adjustment, they do different things to the light that's hitting your specimen. And you'll just have to play with it when we get into lab, but those are actually quite useful as far as helping to see your cells sometimes whenever you are having a hard time seeing them. We will, at some point when we do the motility lab, be looking at the cells when they are not stained at all. Cells when they're not stained at all are clear. So it's really hard to see anything about them without getting contrast somehow, and we will create that contrast. When we're staining, that's what we're doing, right? Creating contrast so that we can see our specimen. But whenever we're looking at unstained cells, you can still create contrast by adjusting how the light is when it hits your specimen. So this is how I'm changing it. But yeah. So yeah, that area where the light is actually shining out through, we're not really talking about that too much, but it's called the aperture. It's not really a term you need to be familiar with. Ones that I might ask you about, well, I could put a sticker on any part of the microscope and expect you to know it, but ones that I often will ask about, light source, I'll put it on the thing down here. Condenser, I'll put it down on thing down here. I usually won't ask about diaphragm because it's hard to separate those two just visually. Condenser, stage, coarse focus, fine focus, arm. And then when we get up to these other ones, right? So the objective lenses. These are the lenses that are actually, now that we've got your specimen sitting on the stage, we've got the light shining through it, and we're ready to look at it and analyze it, right? We start with the lowest one, which is the shortest one. This is the 4x on our microscope. So that means 4 times magnification. Light comes in through here, through your condenser from your light source onto the slide that would be sitting here, glass slide. And then that light goes and hits the lens here on the objective lens. So you're looking at that one, and it's going to give you an image straight away of 4x. So 4 times what the actual specimen is, okay? What you're really looking at. But that isn't what you see out your eye piece up here, okay? Because the light's going to come through here. This lens creates the real image. It's the image you created directly from the specimen. Specimen, lens, real image, because it's directly from the specimen, okay? It's going to hit some mirrors because clearly light doesn't travel like that. Hit some mirrors to get it to this eyepiece up here. This is another lens. I told you about it already. Ours are 10x. So you're going to go through another lens of magnification here. So 10 times more magnification added on to this, but it doesn't add. That's not how it works. So you have 4x here. We have 10x here. So once your specimen light goes through here for 4x, that 4x size is going to hit this lens here to create the virtual image because we went through another lens. So it's kind of like a fake image, right? Another image being created. And that is 10x. So that 4x times the 10x will be what you're viewing. So 40x. So if you're on the 10x and your ocular is 10x, then that would be 100x, right? So 10x and 10x, 100x, 4x, 10x, 40x. I mean, it's not hard. So just basically look at it as, because all of our ocular lenses, and what I will do in the lab exam is ask you how this microscope is set up right now that you're looking at. You would look at it. How is what's the magnification total that you would be looking at? And be nicer wording, of course, but and then you would say, okay, this is on the 40x, and we have an eyepiece of 10x, so that's 400x magnification. Okay. So it's just multiplying, and luckily it's tens, so it's not too bad. It's always going to be tens because our microscopes are all 10x on the eye lens. So easy. Multiplying. So that's how you figure out your total magnification. Just that is something you need to know. And you need to know that for both the lecture portion of the course as well as the lab portion. Anyways, so your light goes through your objective lens, and then goes the mirrors and out the eyepiece, and you could look at it. So we made a whole way through this whole thing. When you put your slide down here, you want to adjust first on your lowest power lens using the coarse focus. You get your image where you can roughly see it. Use the fine focus to fine tune it, right? So now you're looking at something very clear on your lowest power that you adjusted first with the coarse and then with the fine. Okay, so you're ready to see it even more magnified. You shouldn't be just switching straight from 4x to 100x unless you are very adept at using the microscope, and I promise you, you are not. So do not do that with our microscopes when you first start. Just go ahead and switch through all of the lenses. Humor me. Go from 4x to 10x, and once you're done getting it in focus on 4x, it will stay at the same spot. You're looking at the same thing every time. I promise you are. You're zooming in on exactly center. So if you're looking in your microscope and that big old circle and what you're interested in was over here on the side, and you flip over to the next zoom in lens, it's probably going to zoom past what you were looking at. So you might be like, I don't see what I was looking at. You said it would stay in the same place. I promise you it did. Your thing was out on the side. We didn't have it in our field of vision anymore. So get whatever you are looking at centered in the very, very center of that circle So you'll position that using your stage controls, your right and left and forward and back. And then it's centered, you'll switch to 10x. You will not touch that course control from here on out. You don't need it. You only needed it at the 4x. It should be close. So we have a man come in and calibrate the microscopes every semester. I think it's every year for micro, and then it's every other year for physiology. But we use our microscopes a lot. But anyways, they're calibrating them now. We had to pull them out of the freaking asbestos room to get all of them. And the guy's coming and calibrating all of them now. So they will be working in perfect order. When you get them back, they'll all be checked over. So if you are at your 4x and you switch over and you are just completely nowhere near anything clear when you're looking at it on the 10x, you did something wrong, not the microscope. So if it's switch over from 4x to 10x and you look at it, it might be blurry and you might have to adjust it a little bit with your fine focus, but it should be in that general vicinity of clarity, okay? If you're going really far one way or the other, you weren't focused on what you thought you were, okay? A lot of people think that they are focused on something clear on the 4x when they're just looking at just a white blank space. And I don't know how you focus on something like that, but people think they do. So that's usually where your problem starts, is you're not even starting out focused on the right thing. So how can you fix it from then? But yeah, so 10x. Get it clear with the fine focus, all right? Then switch to the 40x. Get it clear with the fine focus. In the 40x, if you're lucky, you might have some bacteria that you can start to see in the 40x a little bit. When you're really looking at it and getting comfortable with it, if it is stained, start to see the cells. A lot of them you can't see until you get to the 100. So so we got it on the 40x and it's clear. So we're ready for the 100. The 100x is our oil immersion lens. And you always use oil with it. It's not one of those things where you switch over, you kind of get it clear and you're like, okay, time for the oil and then put it back. No, if you're going to 100, you better have oil on there. Otherwise you might not be able to even see the specimen there. Sometimes you can, I'm not gonna lie, but put the oil on there, man. We need it for a reason. Put it on there. It is not for lubricating the lens, right? The oil is there in order to basically reduce light scatter. So as the light travels through the glass and then to the lens, if it passes through air, there's a big difference between like the density of air versus the density of glass. When we put the oil on there, it's a much more similar density. So we can maintain that density all the way to the lens. And even though it's going to look to you like your 100x is touching your slide, it isn't. There's space there. So the oil like seals that gap and reduces light scatter and gives you a much clearer image. Much, like worlds different. It's a little finicky on the oil. You want to get a healthy drop on there is what I say, about the size of the light hole that you'll be looking at, okay? Maybe that'll make more sense when we do it, but put the oil on there and then put the lens on and then use the fine focus to fine tune what you're looking at on the 100x. 100x, always use the oil. Oil is only used on the 100x. That's that, okay? Just reduces light scatter and noise. So I think we went through all the parts, and this just explains all the parts that I'd expect you kind of to know and what they do. Okay. All right, so I told you that on your slides that you'll be preparing that you will need to stain them. You can, I want to be clear about this, you can stain slides without heat fixing them, but heat fixing is a process. You can put your slide through, your specimen through. We'll take a colony off of a plate or off of a slant or a drop of broth. Broth is probably easier. My internet connection is unstable, go figure. Broth is probably easier because you can just take your growth in your broth. It's liquid. dip your loop in and just put a drop on there, spread it out and let it air dry. No fussing. If it's coming from a solid surface, you probably need to add a little bit of water. And I mean a very little amount of water. If you are using your little squirt bottle from your deionized water and squirting the water onto your slide and mixing your sample in with that, too much. End of story. Never do that. Okay. Won't ruin it, but you'll be waiting about 5 years for that slide to dry. So, and it does need to dry. It absolutely must. So, you really want to not put very much water. What I do is I take my water bottle and my loop that's clean, and I just squirt a little bit of water onto the loop. Just a drop of it will be in the loop. Then I touch that to my slide. That's all you need. Do not need more water than that. Okay. And then I will sterilize my loop again and then get my little sample off my plate of that one little colony, just touch it in there enough that you can see that it's on the loop, and then just mix it in with the water, like you would imagine, and spread it out on your glass. Let it air dry. Once it's spread out enough, you want to be able to see that there is a film on there, but that's about as thick as you need it. Just a general thin film. It should be translucent, you should be able to see through it, but it'll be cloudy. If it's thick where you can't see through it, guess what? The light can't pass through it either, and you won't see anything. It'll just be black when you look at it in the microscope, and it was a waste of your time. I can salvage most things though, so no worries if you mess up. I'll help you with it. So, but yeah, so nice thin layer, let it air dry, please. Then after it's air dried, you will take it with a pair of little tongs and pass it through the Bunsen burner flame slowly. So like if this is your flame, you'll pass your slide through the actual flame. Like I go, shh, and that's one. And then I'll do that like 3 times, okay? You don't want to hold it in the flame, it will burn your sample. You don't want to put it over the flame when it's wet because it will boil your sample. These things will damage the shape of the cell, the contents of the cell, and certain aspects of the cell you might be trying to see. So that's why it has to be dry, and that's why you want to pass it through the flame, but not hold it there. The goal is to kill the microbes, yes. So heat fixing, it will kill the microbes, adhere your sample to the slide, and it will preserve structures of the cell so that you can see them in the microscope. That's what it will do. And it will make your stain work better. There is a stain, there's at least one stain that we'll do in lab that does not have heat fixing, and there's a reason for it, and we'll talk about it. It's the capsule stain, and we'll get to it when we get to it. But yeah, so we'll heat fix almost all of our slides and then go on with the staining procedure. The first one that we're learning in this lab is the simple stain, and it is easy. It is simple, and please don't forget about it because there might be a time in the lab later on where there's freaking gnat in here. Why? Living near the lake is like bugs 9,000. Yeah. So heat fixing, simple stain is just putting a stain on there to get some contrast. That's it. So you can see the shape of the cells, the arrangement of the cells, and that sort of stuff. So simple stain, after you've done your heat fixed slide, you will just put one stain on there, like methylene blue, and let it sit usually for like a minute, and then rinse it off, dab it. They say blotting paper. We don't have that because we're cheap, so we use just paper towels. And then after it has dried appropriately, which takes little to no time at all, you will look at it in your microscope like we just described. So that's how the simple stain will work. Just provide some contrast so you can see shapes and arrangements of the cells. So having said that, this part doesn't talk about the shapes and arrangements, but when you get into the questions on our part that you would be turning in for credit, this part does talk about the shapes and arrangements. See. So we have all of these terms are here defined out as you would need them, but I'm going to go ahead and go over them in some images because it's easier to understand it that way. So let's go with the cell shapes first. Microbiology. All right. Fine, sure. Okay, so we've got I don't like this though. That's the too much information all at once. I want images. This is better. Okay. All right, this is good. So we have the cockeye, the two biggies, and the only two that you will ever see in our lab exams will be cockeye and bacilli. You are welcome, because the other ones can get confusing. Be aware of them. You might see them in like a quiz or a homework or something, like spirochete or corkscrew or helical, whatever. They're spiral shaped. Vibrio, when you talk about Vibrio cholera, these are comma shaped and whatever. So there's other ones, but the ones that you're like, and Corynebacteria have a club shape. They don't really in our lab, like we will see Corynebacterium, but they look more like Coccobacillus than anything. But anyways, there's others, but the ones I really want you to be familiar with for the test, and the only ones I'm ever going to test you on: cockeye, round; bacilli, rods; cockeye, round; bacilli, rods. Okay, always. And if you hear the name of a bacterium, something coccus, Staphylococcus, right? Coccus, guess what shape its bacteria are? Round. If you hear something that is bacillus, lactobacillus, those are very common, especially we can use Lactobacillus San Francisco for sourdough bread. We can use Lactobacillus bacteria of different species, Acidophilus, for example, in the genital tracts of people in general, but especially in the vagina for women. Important for keeping that acidic pH. So these guys are named based on their characteristics. Easy, that makes it so easy for you, right? So if I were to put on a test, a question that said organisms that are named Lactobacillus, Bacillus anthracis, Lysinobacillus sphaericus, and Staphylococcus aureus, and I had a whole big list of those four, and I said, which of the following bacteria has a spherical or round shape to the cell? Then you should be able to say Staphylococcus, because all the others had bacilli in the name. It does follow that rule. It is that specific, okay? So cocci and bacilli, those are your big shapes of the cells themselves, the actual individual cells, their shape, okay? So let's talk about the arrangements next. That's how these cells can be grouped. Sometimes you'll see them in long chains. Sometimes you'll see them in general irregular groups. And sometimes they'll be stacked up like crazy, or in pairs even. So that's what this is also talking about here on the left of this chart. Diplococci, two round guys stuck together. You see it stuck together like that in an image like that. When you look at an image of Neisseria meningitidis, it is the cause of the worst kind of bacterial meningitis that you can have, the worst kind of meningitis that you can have. And it looks like butts. It's kind of cute, but they are diplococci. And it's very characteristic of Neisseria species to have that. There's another Neisseria species that you should probably be familiar with. All of them have that diplococci look to them, and they're gram-negative. Neisseria gonorrhea. I'm sure you can guess what that causes. So yeah, very characteristic of some of these guys. You can see here below the diplococci, we're talking about the streptococci. Strepto, I remember it like this: strepto versus staphylo, which those are the two that I would ask you about on the exam typically, strepto or staphylo, okay? So you got cocci or bacilli, and you've got strepto versus staphylo. Those are the two biggies. Strepto, like a strip, it's a chain, is what it really means, like a chain of cells. But I think strepto sounds like strip. And staphylo means irregular bunches, or bunches like bunches of grapes, is how you can think of it, okay? So Staphylococcus aureus is going to have coccus, round cells that are bunched up in irregular bunches like grapes. Then we have Streptococcus pyogenes, the cause of strep throat and rheumatic fever and scarlet fever, and necrotizing fasciitis, and all sorts of stuff. So Streptococcus pyogenes would have that strepto appearance, that chain. And it is very characteristic when you look at some of these streptos that they have, you look at them in the microscope, it is chains, and it is so cool to see. But yeah, And then we have sarcina, which is like cube shaped, and then tetrad, which is just in fours, but they're not cubed up in that 3D fashion, right? All right, so for the bacilli, the rods, they can diplo, so they can come in pairs. We can see that sometimes. They can strepto, so we can see that in strips. We don't really see staphylo for the bacilli. They can be just randomly all over the place. Oh yeah, I should say hint, hint. You don't always have to have a characteristic arrangement. Sometimes you can say this bacteria doesn't really have an obvious arrangement. You might have a few of them stuck together in pairs here or there. But man, when you look at Neisseria meningitis, they are pair, always. So it's very characteristic. So bacilli don't really do staphylo, but they can do strepto, they can do strips, they can line up in lines end to end, like it's showing in strepto, or their sides can be sort of paired up. So it's usually their ends that are attached somehow and not really sides attached, but they'll stack up almost like that, a fence. That's called palisades. A palisade is always a fence. It's like a defensive fence, but yeah. So that is the cell shape and the cell arrangement terms that I would be familiar with. They're on that handout that we have. Again, these are available on Canvas in the modules in lab documents and worksheets, or you can find it in Top Hat for each, like if you look at the content and not like what is due and how that's listed. You can find these sheets, and I assign them, quote unquote, for reading. So they should be there available for you to look at. But yeah, that's that for the content for this one. To go over these is for what you guys are going to turn in. I would have had a picture for you guys to take to upload if I had the freaking thing saved. But I didn't. So I'm going to need you guys to fill these out. I don't care if you print them and write on them and take a picture and upload that, or if you use your tablets like you guys like to do, which I fully appreciate, and use that to fill this out. Or if you literally just in a separate document entirely, just go number 1 and put your answer, and number 2 and put your answer, and just, turn it in like that. I don't care, but put answers somehow. I'll kind of go over these with you guys, and I'll give you images to look at that I will also post on the discussion afterwards, so that you have them to look at to actually answer your stuff. But we don't have results, right? So using appropriate terms, describe the bacterial growth on your street plate, and anybody's look different than yours at your table, and why might that be? So we're going to look at it as this is what your quad street plate would look like. Images. See if I can find the ones I was going to use. Yeah. All right, let's say this is what yours looked like. Okay. You see how it's got these kind of off-white colonies here, and they're kind of all round and they're opaque, and whatever you could describe it with your colony morphology and all that, which is what the idea is here. So you would look at your assignment here, appropriate terms, colony morphology terms from the front of the sheet, which is this stuff up here, right? Use those terms, use a few of them, use at least 3, please, to describe what you see on that plate that I just showed you, and I will post that picture, I promise. Okay, but here it is for now. You pick terms from this. And it says, did anybody at your plate look different? Well, I mean, I was going to pick this, let's say, Serratia marcescens. Let's look at Serratia marcescens. Spell it right, by the way. Oh, yeah. Oh, that was weird. I didn't expect it to do that. I didn't know it could. Okay, well, whatever. Here we go. So this is what Serratia marcescens looks like. So let's say you're the other plate that you're looking at. Let's say it's somebody else's plate, like right next to you, or whatever. So the next plate at the table. I know it says anybody else's plate at your table look different. Let's say we're talking about a whole other plate. Okay, that's what I want you to answer this as. So your number 2, compare that first plate to what you see on this plate. Okay, and why might they be different? Like, why does this one look different than yours? You could say, it is as easy as saying, like, it's a different bacteria, so This one maybe makes a red pigment or whatever, okay? If you guys, and you can include the pictures when you're submitting it if you want to, if that helps. I don't really care. And like I said, I'm going to post these on the discussion thing where I've been posting everything. So you guys can have these. It was meant to be in the presentation. But anyways, that will be that question, okay? Or you can be thinking it's just being different plates. If that helps, you don't have to look at them. No, we have the creamy white one with the little colonies and the red one with the shiny, yeah. So it's fine. So do it that way. Just understand that there's different bacteria out there and tell me why they're different and give a general concept. Okay. No big old, don't get into the genetics. I don't need a paragraph here. Why might it be? Different bacteria survive in different environments, so they have different aspects and characteristics to them. All right, so what shape or arrangements would you describe your simple stained sample as exhibiting? And so we didn't have a simple stained sample, so we are going to look at this one. Sure, this one. This is what your simple stain looked like. So you're going to describe your cell shape, which clearly is rods. And I would be sure that you put bacilli. You can put both. You can say bacilli and in parentheses rods or vice versa, but be sure you include bacilli because that's the term I want you to know. You might have some diplo, but mostly this doesn't have an arrangement, right? So you can say some diplo, mostly irregular arrangement or something like that, random arrangement. Make sense? So that's the questions that you would do. I know that there is down here a grand rounds that wants you to go look at other people's colony morphology and cell shapes and arrangements. Don't worry about grand rounds. I'll tell you what to do for each assignment as we need to until we get back in the lab for the grand rounds, but for this one, just don't put anything. You can just draw a line through it if it helps you. I don't care, but we're not doing the grand rounds this time. I do recommend you look online and try to see different examples of these if you can. So, yeah. Let's see. So I'm putting in the chat on this, and you guys can link to it, and I'll probably also link it on when I upload all this stuff. But the chat has a link to my video from like a couple years back, whenever there was a snowstorm and I had to film for this. So yeah, so there's that you guys can go watch. It's probably the similar information, but it's gonna have actual microscopes in it. Can you imagine? So so yeah. Here's this image for your simple stain. I'll leave this up for a little bit and do this one. Where's this one, right? Chat. The other plate. Let's just try to find that Serratia marcescens. I like it because it's so different. Look at it. There's no mistaking that it's different, right? All right. So there in chat should be everything that you need to complete that worksheet. Remember, you don't need to do the grand rounds. I don't want to take up so much time. Just talk a lot, don't I? Okay, well, at least I'm not trying to teach you guys Morse code or something. But yeah, so you made it. I made it. We're done with it. Any questions for anything?
Good afternoon. Good morning, everyone. I have a question. When is our midterm?
Midterm, it is on the syllabus. I don't know off the top of my head, but let's look. All right. Also on Canvas, you'll have like these things that look like this on a lot of the pages. Syllabus, course instructor, class schedule. So all those will have those information. Let's just click on this because it's easier to see. But so we have 6 exams in this class, right? So our first exam for exam 1, chapters 1, 2, 3, 4, 5, and 7. Okay, that's on next Tuesday, the 11th. So be aware of this. So next Tuesday, the 11th, we have 1 through 5 and 7. We will have a review on Monday. So Monday, you're expected to go over chapter 7. Probably everybody will go over all the chapters. On Monday, you do you on whatever time schedule you think you need to do it on. But we will have a review probably at like 8 p.m. on Monday evening. It will be online on Zoom, and we will do a Kahoot group thing. So I might have like a mini kind of go over your study sheet, which has everything you need on it, and review kind of briefly over that. And then we will do like a group game of the Kahoot where we'll all play together, not individually. It's a lot more useful that way, I feel like, and more fun. And it has repetition aspects to it that is more fun too. So we'll do that for the review. For however late you guys end up wanting to stay on for it. And then the exam will be on Tuesday. It will be on campus. And whenever we get to Monday, hopefully by Monday, because I emailed Dr. Rhodes and I'm trying to be sure what room we can be in, because I don't think it's safe in the lab right now. But it might be in Science Building 307. But I'll let you know that we will have an in-person exam, even if it's not in Science Building. So you'll bring your computer or tablet and we will have that first exam. That's just Unit 1 exam. Unit 2 is kind of like a midterm for the lecture. It's going to be Unit 2 plus Unit 1 together. It is comprehensive. It's comprehensive. Exam 3 is not. There is a reason for this. People do not expect the types of questions that I ask on the exam 1, even though I try to prepare people for it. They just don't believe me or something about it. So whenever I tell you guys that some of the questions on the exam are going to be very similar to what you see in critical thinking questions on NCLEX, they are very similar to that. I'm going to give you scenarios, and then you have to answer based on the scenario. If I tell you that there's an example. If I tell you that a man goes to a Chinese restaurant and orders, some fried rice and stuff, and goes home and he gets sick like within 6 hours later. It lasts about 24 hours, but it ends up happening to a whole lot of people that went to that Chinese restaurant. So an epidemiologist goes out there and does some sampling of some stuff and finds out that there are endospores in the rice. Which of the following organisms could have caused the diarrhea associated with what these people got, right? And then I give choices. The choices could be, this isn't one of the questions that's on there, but it's similar to what you might see. Choices might be Bacillus cereus and Salmonella enterica and Escherichia coli, and it could be, I don't know, Staphylococcus aureus. I want to be clear, all of those cause diarrhea. How do you know which diarrhea causing bacteria is causing this issue? Now, if you've read through chapter 4, which today should be a chapter 4 day, or wait, chapter 4 was Wednesday. So you should have already been through chapter 4. If you read through chapter 4, then you would know by now that when we talk about endospore forming bacteria, there are only 2 genuses of bacteria. So that first name in that binomial nomenclature that we're talking about, the first name is the genus. The second name is the specific epithet. The first name, Bacillus or Staphylococcus or whatever it was I was saying, only 2 genuses make endospores ever. And that is Clostridium, which I didn't name any of those, and Bacillus. Okay, so that's how you would know that it's Bacillus cereus. It has to be because none of the other ones make endospores. Those are the types of questions you're going to see on the exam. If you understand just simply that endospore forming bacteria are only Bacillus and Clostridium, which is in the notes. If you understand that, that's the only information that you needed to answer that question. But people see diarrhea and they're like, oh, salmonella every time. So often people get questions like that. It's not meant to trip you up. You're just meant to understand, remember that, okay, what are the key things here? Because whenever you get into nursing or medicine or wherever it is that you're going after this, it's not about remembering bacteria give you diarrhea. It's about knowing the difference between them. So this is testing you on that knowledge. I do not expect you, there will be questions on the exam that are just regurgitating knowledge. Of course, I have some like that, but there are other questions on the exam that expect you to actually use that knowledge. That's where people get tripped up on this test, okay? People do not do well on the first exam because they expect it to be regurgitate the definitions. It is not. So it will be questions like that. So you will have scenarios and you will have to apply it. And everybody will hopefully do well if now that you've been warned, right? So you're being warned. I don't bring it up as a surprise to people and just say, good luck. I warn people that it is going to be a difficult test and you have to be able to apply, and I give them an example and stuff like that. So just be prepared for that. Yes. So people don't do well on it. So that's why exam 2 is a comprehensive exam, because if you do very poorly on exam 1 and you do better on exam 2 that has some of the same material, I will replace your exam 1 grade with your exam 2 grade. Do not rely on that though, because people usually do worse in general on exam 2 than they do on exam 1, because it is a lot more new material, because it's genetics, because we're going to talk about DNA and how DNA replicates, and we're going to be talking about genetic engineering. We're going to talk about metabolism. This is stuff that people have usually never learned anything about at all. And so it's more difficult information. Whereas chapters 1 through 7, if you think that that's the difficult, complicated information, I hate to break it to you, that's just the platform we're building from. So, yes, I don't expect you to know details of the chemistry of metabolism. We're not going to go over every single chemical step of the Krebs cycle or something like that. But I do expect you to know what the Krebs cycle is. I do expect you to know what is going into the Krebs cycle and what is coming out, generally speaking. You know, and what it's for, and what is that? Where does it happen? The concepts of it. So, but if I'm saying Krebs cycle to you and you don't know what I'm talking about, unit 2 is going to be a rough one. So just hold on tight to that. But yeah, if you can do better on unit 2 than you did on unit 1, better on unit 2 than on unit 1, then I will replace your unit 1 with your unit 2 grade. People do better. I'm not saying they never do. It's just, it tends to be more challenging. I should say that. It's not necessarily people do worse. I say it's more challenging. That's the better way to put it. But yeah, if you can step up to the challenge, then you ain't gonna have a problem. It's about learning how to study, and do not depend on 2 hours of study time before the exam to get you through these exams. You won't do well. People a lot of times think I'm just shooting fire out of my ass for no reason, but this is a very difficult course. So usually people haven't seen this stuff before. So that's why I'm warning you guys. And if I warn people, they tend to do better. So this is just me being honest. So the lab midterm, so that one is coming up on June 25th. So the first lab test will be the midterm for the lab, June 25th. So the lecture has tests and the lab has tests, lab midterm, June 25th. So we have 19 days, literally, until we get to that. We are at this Thursday, so we've made it through this first week. So that's all been virtual. If it's all been virtual, and we literally get to that midterm somehow without ever having actually seen a microscope or acted on a microscope, then your test will not be using microscopes. I will teach you how to use a microscope. I will teach you how to use a microscope in person, it will happen at some point, but it has to happen in a safe environment where you can actually use it. So I'm not going to test you on using that microscope unless you've used it. So if we get to the midterm and it has to be on paper only, then it will be on paper only. We'll just use pictures and it's fine, and we'll do that. I'm just really hoping we don't have to, because that's going to suck like bad. I know there's a lot, but I hope that answered your question. Any other questions, guys?
No, ma'am. Thank you.
Thank you guys so much for showing up today. I think I really did cover pretty much everything you would have seen anyways in that other one, but as soon as I have access to it, I will post it. But yeah, I'll see you guys next time. Be sure you're getting as much studying as you can. I know it's Thursday, but don't just mean that shut off completely. Try to put a little bit of time in these next couple of days to study, because we do have that lecture exam on Tuesday, and man, did it come up quick. I am, yeah, it really did. Yeah. So to put the effort in, we'll have the review on Monday. So hopefully you guys will all be ready, and we'll all be ready to review on Monday evening. Yeah. So I'll see you then. Yeah. Okay. All right. Good job, guys.
All right. See you later.
All right. Thanks, everybody. I'll see you next time.
All right. Bye-bye.